Okay, hi everyone, welcome back. I'm glad to see people come for the second day of our RNA-Seq tutorial. Before we dive into writing more shell scripts, Dr. Adriana Guy from the Genome Technology Center is going to come and give you a little introduction to the molecular biology and details of preparing samples for RNA-Seq. I just wanted to say that if you're planning to do RNA-Seq experiments, you definitely want to have a conversation with her before you walk into the lab with little tubes of RNA. Hopefully you have that conversation while you're writing the grant that will fund the future experiments. Okay, Adriana.
All right, thanks Stuart for the introduction. So Stuart told me to keep this brief, so this is going to be very general, but if you have questions, please interrupt me, it's no problem. I would like to also emphasize that it's very important that we talk beforehand because RNA-Seq library preps became this huge thing where there's many different kits, many different ways of doing RNA-Seq, depends on your purpose, what you want to see, what your hypothesis is, and also importantly what your budget is, because some of these RNA-Seq modalities are much more expensive than others. So very briefly, what is RNA-Seq? The first thing I always like to say about RNA-Seq is that RNA-Seq is a misnomer because we're not really sequencing RNA. We are isolating all the RNA and converting it to cDNA, and what we're actually sequencing is DNA. There will be future technologies, one of which is Oxford Nanopore Technologies, in which we'll be able to sequence the RNA transcripts directly, but this is something into the future. But we are going to have this instrument in-house. But we're talking maybe a year or two down the line, but I'm just raising the awareness that that's something that's going to be happening into the future. And then, of course, Stuart is going to be showing you what to do with all those sequences of cDNA that you get and how to map them back to the genome. So it's a very comprehensive and quantitative view of the entire transcriptome, if you want, depending on the technology, but we have to be aware of a few challenges that have to do with RNA-Seq. So one of them is that until we are actually sequencing the RNA, we are basically sequencing cDNA, and there could be some cDNA artifacts. So basically, you're going to be mapping back all those sequences to the genome and to the transcriptome, but mapping reads to the genome is more difficult because in the genome, your reads that are going to be only in the exons are going to be separated by the introns. So that, creates an additional challenge. The other issue is that the abundance of RNAs within one cell vary by several orders of magnitude, and you can have transcripts, for example, for TERT and other important genes, in which you may have maybe 10 copies per cell, and other transcripts in you're going to have hundreds of thousands of copies per cell. So as you can imagine, what's going to happen, because the sequencing, you're just randomly sampling what you have there, that the genes that are going to be very highly expressed are the ones that are going to soak up the vast majority of your reads. And so if you're interested in looking at transcripts that are very, very low abundance, you're going to have to sequence very, very deeply, get a higher number of sequencing reads to look at those transcripts, or we're going to need to use some specific target capture to look specifically at those subsets of low abundance RNAs that you have and ignore all the rest. But even if you're looking at high abundance RNAs, one of the things that you first need to do is eliminate ribosomal RNA. And sometimes mitochondrial RNA, that's not such a big problem. But ribosomal RNA, we definitely have to eliminate and make sure we don't sequence, because that's going to be the majority of the RNA in any cell. The other challenge is for those of you working on microRNAs and other small RNAs, that requires a different library prep, and I'm going to briefly explain to you why. And also the bioinformatics are quite different, so I don't think you're going to address that in this particular case. So the most common type of library prep is doing PolyA selection. And that's because when you're selecting on PolyA, you're only capturing mRNAs and some long non-coding RNAs that have a PolyA tail. And in this way, you can already not worry about ribosomal RNA at all because that's not going to be primed. And so typically the most typical RNA-seq library prep based on that is beforehand you capture just in a regular way that you would do by any PolyA, like for doing qPCR or for doing any other type of cDNA procedure. And then we fragment the RNA and we prime it. And then a first DNA-cDNA synthesis occurs and second strand cDNA synthesis. This is similar to all the procedures that you do in the lab. And then at that point, we have to repair the ends of the molecule and add an additional A. This is traditional in all Illumina preps. For preps with other machines, it's a little bit different. And then after that, we have to add the DNA sequencing adapters. Those adapters are called the Y adapters because of the way they're done. There's a mispaired area at the end that is going to become paired when you do the PCR amplification, which is the last step of your library. And those adapters contain also barcodes or index sequences so that you can multiplex more than one sample per lane, because otherwise you're going to be spending a lot of money and your PI is going to be very unhappy. So basically in Illumina, you end up doing 2 reads, or you can choose to do only 1 read. So it could be single read or paired end. And in addition, there's also an index sequencing read, which identify which sample the reads came from when you're multiplexing different reads. One word about PCR amplification is that because you cannot avoid with RNA-seq PCR amplification, the higher the number of cycles, the higher the number of PCR duplicates, and I think you're going to cover that at some point. No, not much. In any way, PCR duplicates are basically an artifact, and you want to somehow account for them depending on the modality of sequencing, but just to bear in mind that it's highly dependent on the number of PCR cycles. So very recently, strand-specific RNA-seq library preps became very popular. And why are they so popular? Because basically at that point, you know exactly which strand your RNA is coming from. In genomes like human and the mouse, you don't have a lot of overlaps, but you do sometimes, like in long non-coding RNAs, maybe coming from like a different strand than the coding RNAs, and you want to make sure that you capture that information. But if you work with C. elegans, Drosophila, or bacteria, this is critical because there's a lot of overlaps, and if you don't know from which strand the transcript is coming from, then you're going to confuse one transcript with the other and you won't be able to quantify. And there are methods bioinformatically to do the analysis strand-specific versus non-strand-specific. So in this case, what we're illustrating right here is a method to use the ribosomal RNA depletion, which is just basically a reagent that soaks up all the ribosomal RNA and you get rid of it. And then after that, once again, you need to fragment. The need to fragment is because the clusters in the instrument that originate the reads need to be a certain size for Illumina. For long reads, preps, like the Oxford nanopore I was referring to, and so on and so forth, fragmentation is not necessary. And so the middle method, the one by the arrow, there's several different methods. That's sort of the one that came up being the winner. So how do we know from which strand it's coming from? Well, basically, after random priming, and the reason to use random priming is at this point you have the entire transcriptome. Any transcript that is bigger than 200 nucleotides you will detect by using this technology. And at that point, when you are doing your cDNA process, you're going to be incorporating DUTP. And then after ligation, you're going to get degradation of the DUTP. And then when you do it by PCR, then you will basically be making a library from exactly that strand where the transcript originated from. And this information is important, like I said, for small compacted genomes. So this is like our second most popular. It's a little bit more expensive because of the RNA depletion. However, there's a method to do strand-specific RNA-seq using the PolyA tail, and that's just actually just as expensive or just as cheap as the original one. So we have basically switched all our preps to strand-specific unless we are just accumulating samples for a very old project, because you get additional information and it's for the same money. So we are doing all of our preps in a strand-specific fashion nowadays. And just an example from NYU, I don't want to dwell on it, but by looking at the entire transcriptome, you may end up discovering new entities like this specific long non-coding RNA here from the iFantas lab that a graduate student who just graduated discovered. Now, RNA is not always, it's not always possible to obtain RNA in very beautiful shape and form or high quantities. So those are what we call the difficult RNA samples. We can work with all kinds of RNA samples. For example, low input. So the low input RNA samples, we have two favorite library kits. One is the ultra low smarter kit from CloneTech that can go down to 150 picograms of input RNA. There's also the NuGen Ovation, which we like very much, that goes from 500 picograms, 50 nanograms. And I had an issue with the slides right before coming here, so that's why you don't see that one in the slide. Basically, it's your choice which one to use. The only caveat is if you are working, once again, with non-mammalian species, the smarter kit does not work, and we will have to use the NuGen kit for that. And so that's a similar workflow to the other RNAs, but I just want to emphasize that it requires an additional step of amplification of the cDNA because we start with such low amounts. This also gives good results. So for example, this is an example that we started with cells that were sorted. So it was a small population of stem cells from a tumor, and we used the ultra low input RNA-Seq, and we did indeed identify a molecular signature that could differentiate these two populations of stem cells. So that's just an example that I wanted to show it's possible. This has been written up by a paper. You get really good data. So the latest thing is also doing RNA-Seq on single cells. So why would you bother to do RNA-Seq from single cells? Well, if you look at the graph there, you can see that if you have two populations of cells within your cells, some of them may be expressing the RNA at high level and some at low level. And if you're looking at it in bulk, then you might get the wrong impression. So for example, on the right-hand side, you would see for gene A and gene B, if gene A and gene B are fully correlated, then it doesn't really matter if you're doing the bulk RNA-Seq because it will look the same. But if they are anti-correlated in the different populations, then you would get completely different results. And this is a good way to identify subpopulations of cells in a heterogeneous cell population. And one of the most popular and pioneer methods of doing single-cell RNA-Seq is using the fluidine system. There's a whole bunch of other systems over here. That also is a very competitive area, and there's new systems coming out this year. We do have the fluidine system, which basically uses these butterfly microfluidic system to trap one cell per chamber. And at that point, you use also the smarter kit that I was talking to you guys before to do more cDNA amplification. And if you guys are interested in this kind of projects, we need to coordinate with a flow cytometry or any other facility that is sorting your cell before doing this. And you can do 96 cells or you can do 800. And here's an example of a clustering method in which you can see two populations of cells, the blue and the red, which were already separated. But you can see that by using this method, we identified some new subpopulations of cells that were not known before based on their transcriptome, and that they, in this particular case, made biological sense. Just one word about small RNA-Seq library prep, since Stuart is not going to cover this. Small RNAs are too small to be sequenced by Illumina without the addition of different adapters. So basically, what is done is the adapters are ligated straight into the mRNA, and then at that point you continue. You basically are making your miRNA longer than it should be by these extra adapters, and then you build another library. It's quite more complicated and quite more expensive, but it's also doable, and it also requires very specific bioinformatics. That's basically all I have to say. Any questions? All right. Thanks.
I have a question.
Yes.
Regarding bacterial RNA, you give them PolyA tails.
You mean artificially? If you want to add PolyA tails artificially to your bacterial RNA? Some people do that, but in general for bacterial RNA, what we do is we add, we use a special ribosomal RNA depletion, which is actually called epidemiology ribo0. The reason why it's called epidemiology is because people can take samples that are mixed mammalian and bacteria, so like a host and an infected, and then you can get actually reads from both the bacteria and the host at the same time. So that's actually quite a popular way of doing it. Any other questions? Yep.
As for stranded sequencing, like is the cost much more, I expect, because you have to sequence both ways, right?
Actually, no. You can sequence just single strand because the method itself, what it does is it will identify that every read you get was coming from the original strand that originated the message, because you're using the DUTP and then getting rid of that strand. So actually the price for PolyA stranded and non-stranded is the same, which is why we completely switched to stranded. For using the ribodepletion, the method is actually only exclusively stranded, because since you're sequencing the whole transcriptome, otherwise it's really messy to map things back to the transcriptome or to the genome. All right, thank you.
All right, thank you, Adriana. So I'm batting cleanup for this workshop. The last section is how to do analysis of the data that you receive back from the sequencing lab. And I'm going to review some things that Adriana just said and a fair bit that was said yesterday, and I'm going to go really quickly. So if there's a question coming up, stop me and we'll go over it and make sure that you're clear. The whole point of working up to this is because I want to bang out and get to some shell scripts and have you know what I'm talking about. Right, so this is what I'm going to cover. We're going to review quickly the shell scripts, review super quickly my favorite Linux commands, environment modules, batch jobs, all from yesterday. Then with some new stuff, reference genomes on the cluster, the basic RNA-Seq workflow, and I'm going to talk about these programs, FASTQC, TopHat, Cufflinks, and CuffDiff. And then creating SGE scripts for you, and then I'm going to have you do it yourself. I'm going to show you some SGE scripts, and then I want as much as possible for you to type them yourself, submit them into the queue, experience all the errors that you're going to get, and troubleshoot your scripts, because that's how you really have mastery of it and walk away from the workshop actually confident that you can do this for yourself, rather than just grab my script and rerun it and say, yes, I can type the command qsub. I want you to actually feel like you could draft one of these scripts and run this software for yourself. We made a web page for this tutorial. Not sure what's there. We're going to post a lot more this afternoon. So right now, looks like we just have the slides from yesterday and the slides from today. And by the end of the day or Monday, I'm also going to post a couple of papers, some handouts, and a book chapter. So this is the Chibi documentation website, and today's slides are there if you're one of those people who likes to follow along and be able to page back and forth among the slides while I'm plowing ahead. So the URL for this thing is here. So it's https genome.med.nyu.edu HPCF wiki RNA-Seq _tutorial _2016. Those aren't so visible on the link the way PowerPoint shows it. So there were requests for the slides, so rather than email them to everybody, we can put them here, and those who want them can grab them. Right, so we're going to talk about RNA-Seq now. Gene expression is measured by counting RNA in a population of cells. This historically was done using a northern blot. Initially, you would put all the RNA from your sample into a gel and then hybridize it with some sort of a probe and then guess based on how dark a spot that probe made how much of the target gene was present. This technology evolved into a microarray which reversed things. You put your probe into a gel or on a filter. You hybridized it with labeled RNA from various samples, and you measured somewhat more precisely how bright each spot on your array glowed with the RNA from different samples. And then you did some type of a differential measurement to measure the difference in abundance of the target gene in each of your samples. RNA-Seq measures the abundance of gene expression by actually counting molecules of RNA in each sample. The counting is by sampling individual molecules from a library. You're not counting the entire set of RNA, but you're hopefully counting a representative population of molecules from the total RNA that you isolate from your target cells. So we call that transcriptome. RNA-Seq is considered superior to microarrays, which is probably why all of you are sitting here, because it has a broader dynamic range. You can measure differences between highly expressed genes more accurately rather than hitting a plateau. It's more sensitive. You can measure genes that are expressed at a lower level that's sort of at a background or hard to measure accurately level by hybridization. It correlates more closely with qPCR, which is considered the gold standard for measuring expression. But unlike qPCR, it measures all the genes at once, rather than measuring them one at a time. It also has additional information that's not easily detected with an array, such as measuring transcript isoforms, discovery of novel genes, gene allele-specific expression. If you have a heterozygous sample, or you have different samples that are expressing different alleles of the same gene, you actually have sequences of the transcripts, and bioinformatically you can tell them apart. Not all of that information is revealed all at once with just one computational approach. You have to go back and dig into the data again and again to answer different questions, but you don't typically have that same data file, and you can explore it with appropriate bioinformatics tools. Today's workshop is extremely simplified, and I'm just going to take you through one very basic workflow, which hopefully empowers you to start exploring your data, and from there you could choose to learn more advanced computational methods or engage in a more well-educated conversation with bioinformatics experts who you recruit to help you work on your data. So that's our goals for today. Right, so I've simplified Adriana's diagram somewhat to say there's a way that you get your samples from your target tissues into tubes. Then you follow some sample prep protocol. This is one example and a slide I took from Illumina of their TruSeq RNA protocol. It's probably a couple of years old compared to what Adriana just showed. But you get the general idea that we're making a library of cDNA that's derived from the total RNA of our samples. We're using this PolyA step at the beginning in this protocol so that we're only looking at RNA that derives from messenger RNA, which allows us to ignore the ribosomal RNA that would otherwise tend to dominate our sample and force us to sort of ignore a lot of reads that we just paid good money to run through the sequencer. After your sample is prepared, fragmented, loaded onto the Illumina sequencer, it's delivered to you or us, the informatics group, as a set of files in a format called FASTQ, at which point we do an alignment. We match each sequence to the appropriate location on a reference genome. So obviously we need a reference genome for the organism that's being sequenced. There are bioinformatic methods for cobbling together a draft genome for an organism that's never been studied before. I'm not going to describe how we do that, but you do see papers like that sometimes. The draft transcriptome of such and such a beetle or bug that's never before been described. But typically we have a draft genome, human, mouse, Drosophila, C. elegans, and we go through a mapping algorithm to find the corresponding location on the genome where the gene has been transcribed to make that RNA molecule. And that's actually most of what today's talk is about, is that mapping step. All right, this is the data file in what we call FASTQ format. You'll notice that it is somewhat human readable in that it has letters. There are 4 lines in the file for each read that is produced from the sequencer, which typically corresponds to each molecule in your library, which attached to a spot on the sequencing plate and generated good quality fluorescence data. The first line is a header line. We sometimes call that the metadata. It looks very much the same for each read because the first little bit is the name of the sequencer, and then there's a code for the plate that you've loaded onto the machine, and then there's a code for the actual position on the plate of this little spot that's generating the fluorescent signal that led to this sequence. The lengths are all the same because that's the way the Illumina system works. It records one base from every spot on the plate with each cycle of the machine. Then you have the actual sequence of this read. We call them reads. It's a molecule, but it's read for, in this case, 152 bases. And it's in a GATC alphabet. Also, they use an N when there's a base that has not been accurately determined according to their own internal metrics. N's help because they avoid frame shifts. It's like, we didn't get a base on this cycle. We can't tell the difference in the signal between GAT or C, so we write an N. Good sequences don't typically have much in the way of N's. Seems like all of these have an N in the first position, so there might have been some sort of artifact involving a primer, or the way the library was prepared. For some reason, there was a tail of a restriction enzyme, and every every molecule had the same starting base, which will confuse the system since it's expecting roughly 25%. When you take all the different positions into account, all the different molecules, they shouldn't all start with A. So in any case, there's some N's, then there's some bases. Then the header line repeats. In a lot of the more recent FASTQ files, they don't bother repeating the header line. They just include this little plus symbol and a carriage return. Then comes a quality string that seems to be typed in gibberish. That gibberish is known as ASCII characters. It represents a quality score in a range from 0 to 64, where 0 is not good and 64 is really good. In particular, we usually use a quality threshold of 30, which is actually 10 to the third. It's, well, I won't go through the formula, but it's a 1 in 1,000 error rate. So we believe that the score of this base is correct. It's a G, A, T, or C, and we believe that the chance of error is only 1 in 1,000. That's what we call a Q30. A Q20 would be a 1 in 100 error rate. You might think that 99% certainty of correct base is good enough, but for some applications, such as calling mutations in a cancer sample, 1 error every 100 letters would be a problem. It would require a lot more data to sort out the true mutations from those that are just generated by chance. So that's all I'm going to say about FASTQ, except that that's the data that you're going to be working with today, and that's the data that you would typically receive from the sequencing lab, either if you work with the GTC here, or you work with a collaborator, or you downloaded a file from a public database, or you sent out your sample to a commercial sequencing lab. FASTQ is the sort of universal file format for communicating raw results from DNA sequencing experiments. This is another Illumina slide showing that they think that their RNA-Seq gene expression values correlate closely with qPCR. So accuracy and sensitivity for RNA-Seq. So this is what we're going to go through today. We're starting with the reads in FASTQ format. We're going to do an alignment to a reference genome so that the reads line up at a given position over the gene that they were transcribed from. Then we're going to count how deep the reads are over the known position of every gene. In order to do that, we're going to need an annotation file. We're going to need to know where each gene is located on the genome. And then after we do some counting, we're going to compare treated versus control, yellow mice versus white mice or whatever. Typically, you would do this with several replicates. The more, the better. Today's exercise is simplified. We're just going to use 2 files, one for treatment and one for control, or treatment A versus treatment B in this case. In which case, we're going to get a list of genes that are differentially expressed, which will include a gene name that comes from the annotation file, a fold change value, and a statistical value, both a p-value and a multi-test corrected FDR value. Those aren't going to be particularly accurate when you'd have no replicates, but with a better experimental design, you have a moderate amount of trust in those corrected p-values to say, yes, this gene is differentially expressed, it has a fold change of this, and I have a confidence that that fold change is significant at this level. And of course, there's a lot of different statistical packages that compute those statistical certainties in different ways, and I'm not going to get into that debate today. We only have 2 hours. So instead, we're going to use the most popular tool called Cufflinks. And I'm not even going to explain the statistical model for Cufflinks. Instead, I'll just show you how to read the paper. I'm also going to briefly, depending on how quickly I talk, show you how to visualize your data in a genome viewer called IGV, Integrative Genome Viewer, which is produced by the Broad Institute. The nice thing about the IGV is if you zoom in, you can see each individual read as one of these little color-coded lines. In this particular view, they're blue and yellow, indicating reads on the forward strand and reads on the reverse strand. There's a relatively equal mix of forward and reverse because this sample was generated with a non-strand-specific protocol, so the strands were randomized when the adapters were ligated on. So it's not really that the RNA comes randomly from both strands of the genome. It's just that the reads themselves have been randomized as to which direction we clone them. But what's nice about this is if you get a differentially expressed gene showing up in your statistical test, you can go back, load your FASTQ, actually your aligned files, they're called BAM, I'll get to that later, load your aligned files into the genome viewer, zoom in on the gene that was described as differential, load both samples and see if there really is a difference between those two samples that satisfies you on sort of a personal aesthetic level that yes, this gene is differentially expressed at a functionally meaningful way, even if the fold change is 1.1 and the p-value is 0.04. So sometimes the statistics can emphasize changes that are not biologically meaningful, and this is a little bit of a reality test to see if it means something when you look at the actual data. And so it's very nice to visualize your data for some of the genes, as many as you have patience for. Okay, so the informatics workflow that we would typically follow, a simplified version, is to check the error rate. You might filter out ribosomal RNA that somehow snuck into your data. I'm not going to do that today. Align to the genome with a splice junction aware aligner. I'll talk about that in a second. Sum the reads for each gene, which means you need to set the boundaries for each gene. Measure differential expression. If you want, and you have annotation data that describes alternately spliced exons or alternatively spliced isoforms, you could compare those. You could also dig into sequence variants in your transcriptome. I'm not going to do either one of these things in today's workflow. You've seen this slide before. It's important. RNA-Seq is real sizable data. These FASTQ files are up to a gig. If you were going to run this on your laptop, you'd be looking at many hours or days of compute time for each step in the analysis. In particular, the alignment is very compute heavy. The counting and differential expression is moderately compute heavy. So it can't realistically be done in an efficient way on a laptop or even a moderately beefy workstation that you have in your lab. You need some compute power. We've got a couple ways to go with that. You could rent a computer on the internet via the Amazon cloud or some other cloud computing resource, and there are workshops that emphasize using that method. Here at the medical center, we have a high-performance computing system where you can access significant iron, significant amount of CPUs with lots of RAM, and get through an RNA-seq analysis in a few hours with relatively large number of data files. In fact, because we have a batch system, you could submit separate jobs for each sample. Each FASTQ file that's generated, run them all in parallel. So what might take 8 or 16 or 32 CPUs, you could run 8 or 16 simultaneous jobs all over the cluster. depending a little bit on how busy it is. We can't really have 60 people all submitting 16 jobs, all using 32 CPUs, all at the same minute. But we figure your samples will get done on different days. You run your processes at different times, and the cluster can support many people submitting fairly large jobs randomly all different times. As we recall, when we log in, we're going to use the head node, but this is not a job you can run on the head node. You have to write your command into an SGE script and then submit it to the worker nodes via qsub command. Okay, and here's your user's guide if you don't remember all the details on how to write your qsub scripts, but I'm going to review it today. All right, so you could do this now so that we're all ready and to go when I start showing the examples. You want to log in to Phoenix. We're using this key pair authentication, which Eric and Ali have already taken you through. Let's go. Do it. Log in. All right, we're going to write some SGE scripts today. We're going to start with quality control check, then we're going to do alignment, then we're going to do Cufflinks expression measurement. This is just a fine point. We don't have a lot of data files today, but if you did have a lot of data files, you can write a little loop that submits multiple jobs to the cluster for each of your different data files. And I'm going to show you a little bit how to write an SGE script that could accept different commands, different files as input, so that you don't have to rewrite your script each time for each different data file that you're going to analyze. We're not going to run loops today, but I'll show you the structure that would support that.
One point about the loop sub, it's preferable rather than doing a loop that does separate qsubs to do an array job. Unfortunately, we didn't cover array.
Right, that's an advanced topic. Use the user's guide or meet with these guys and learn how to move up to the next level of complexity. I'm not showing you how to do that. I'm just telling you that it's possible. Right, so these are the commands. We saw this yesterday. We're going to qsub to submit our jobs. We're going to qstat to see how our jobs are doing. We're going to qdel if we make a mistake and the job doesn't die instantly, which it probably will if you make a mistake. And we're not going to use qlogin today. All right. We also talked about modules. All the software that we're going to use today requires several module load commands. These are not things that are always running and always available. You have to load them and all of their nested prerequisites. All of these things have multiple prerequisites that they call on. Each program calls subprograms in order to do its work. Specific set of supporting libraries, etc. And we use environment modules. We're going to use module avail and module load mostly today. And as we learned yesterday, it's necessary to put these module load commands into your script. When you're typing interactively, you've got a login environment, but when you submit the script, it's running on a different computer, and the customizations of your working environment that you set up are no longer in force. You have to re-establish the working environment on the subnode, so you need to put your module commands in your script. One important module that we didn't go over yesterday is called iGenomes. I mentioned that in your alignment, you're going to take each read and map it to a reference genome. It's possible, following the instructions for the software that we're going to use, for you to download a reference genome from GenBank, UCSC, Ensemble, Flybase, or whatever, index it, and use that reference genome in your personal account. But with 700 users and each person working with multiple species over time, that would be an enormous waste of data storage and also of the computing that's necessary to create these index files that are used by the alignment software. So rather than have everyone do that and teach you all how to compute your own indexes, we've created a resource called iGenomes that contains reference genomes already indexed in most of the common formats that you need. Everything that you need for this workshop is present in the iGenome section of our server. So, and you don't copy those reference genomes, you just point to them in your script. And I'm going to show you how to do that. So it's a module. You just type module load iGenomes. You can do that. It's really a good idea if you do. What does iGenomes do? Actually, it does only a little thing. It creates a variable name called $iGenomes root, capital, underscore, etc. And you need to use that name in your scripts. $iGenomes root. You're going to see it a bunch of times in the next few slides. It points to the reference genome directory. There's a lot of subdirectories that go by species and then by maybe ensemble or GenBank or UCSC, and then there's a separate folder for the annotations versus the genome. There's a separate folder for bowtie versus BWA indexing. And you just need to know. You need to know either by listening to this workshop about how to use TopHat, or you need to read carefully the documentation for whatever software you're using to do your mapping. It's mostly the mapping, but also the gene counting, because the gene counting requires a reference that locates each gene on the genome. And obviously that is specific to what build or version of the genome you're using. HG19, HG18, whatever they call it now, GCRC36, or mouse, MM9, MM8, etc. Yes?
How do we know what is loaded in iGenomes?
I'm going to show you. That's what we're coming to. Actually, I might break out here and, well, we'll do it in a little bit. When I start writing the scripts, I'll show you how to take a tour of iGenomes just from your laptop so you can see what's there. Yeah, okay, so this is just describing what we're going to do. First tool I'm going to talk about, and you can run for yourselves. It's not a huge success-fail thing. You can also come back and work at this tomorrow and the next day and when you want to dust this off and really get good at it. But the first tool is called FASTQC. It's a quality control tool for FASTQ files. That means it looks at the reads and the quality scores of the reads, but it doesn't pay attention to information about the genome because it's reading just the raw reads before they've been mapped. There are a lot of other tools that do more sophisticated quality assessments of the reads after they've been mapped, but I'm not going to devote time in today's tutorial to them. They mostly, well, yeah, I'm just not going to spend the time. Just be aware that these tools exist and you could learn them yourselves, or you could take another bioinformatics class or meet with a bioinformatics consultant and learn how to do more in-depth quality analysis on your RNA-Seq runs. But if you want to run FASTQC, we do module load FASTQC. Don't run this like this. I'm going to show you how to put it into a shell script. I don't want you all running it interactively, but this is what you would do interactively. If you would say log into a qlogin shell, you could do this. FASTQC minus O dot means put the output file right here where I am currently sitting. And then you just give it the name of a FASTQ file located presumably in your directory, or in this case, in the tutorials directory. And it runs its QC analysis and it generates its output as HTML. It would then write a report.html file locally in your current directory where you submitted this job. And you could bring that HTML file to your desktop using SCP or WinSCP or Fugu or FileZilla, whatever system you've established for moving files back and forth between your current laptop and the server. Right, so, and then this may work. It works on Macs if when you logged in originally, instead of typing SSH, you type SSH minus capital X. It's possible to remotely launch the Firefox browser and view the HTML in sort of an X Windows remote session. I'm not going to try and troubleshoot that for every one of you because it tends to be challenging. But if it's something you want to learn to do, you give it a try. If it doesn't work, you look in the user's guide, try and troubleshoot yourself, then send a question to HPC admins, and they'll try and figure it out for you. If that doesn't work, bring your laptop over to Chibi and somebody will sort it for you. Right, so if you run FASTQC, you typically get an output that looks like this. Actually, there's a whole series of different graphs, but this is the one that people spend the most time looking at. All it's doing is it's taking each base of your read. You know, I showed you a 152 base read. This one is mapping a 250 base read. And it's for each base, it's showing you a bar graph. It's showing the median, and I think it's the median, the 25th and 75th percentile of quality scores, and the range of outliers. I mentioned before that we typically use 30 as the cutoff for good quality sequencing. So if the majority of the bases in all of the reads are above Q30 at that position, then you could safely say that's a good quality read. If in the case of this one, they improve up to a certain length and then they start getting much worse, you might ascertain that something went wrong with the sequencing after about 150 or 160 bases in. There are tools that would allow you to trim away the low quality sequence so your reads become shorter, but they have fewer bad quality letters in them, which might improve the mapping to the reference genome, since bad quality bases might lead to a poor quality mapping or even prevent the read from mapping at all once it hits too many mismatches. So this is a pretty good quality read that tapers off at the end, and it could be saved by some judicious trimming. You could either trim them all to 150 so they're all the same length, or there's software tools that trim from one end until the quality of a window, maybe 10 or 20 bases, reaches some average level. All right, I've talked about the mapping. We're going to use a tool called TopHat to do the mapping. You might know something about sequence alignment. You take one string of letters and you match it to a second string, and you slide them back and forth. You introduce gaps, and you allow a certain amount of mismatches, and you find the optimal frame of alignment. And then you count the number of matching letters, and you derive a score, and you say whether that alignment is good enough. Does it meet a certain threshold of quality? If yes, then we call that the optimal alignment for that sequence on the second sequence. The problem with aligning RNA-Seq reads onto the genome is obviously the introns. The introns have been spliced out of the RNA, and so when you try and match the RNA read onto the genome, you come to a large region of intron sequence in the genome where the RNA fails to match. You need to create a giant gap of unknown size and figure out where the alignment picks up on the other side of the intron. As Adriana mentioned, genes that have tiny exons and big introns create a even bigger challenge for the alignment algorithm. Classic alignment method like Smith-Waterman or even BLAST is not designed to accommodate big insertions, big gaps. It gives the big gapped alignment a poor score compared to a continuous alignment. Whereas in RNA, a big gapped alignment is what you expect and should not be penalized in the score. Even if there are several big gaps in one RNA read compared to the genome, that's perfectly acceptable. Now, another problem is we have millions of reads. 40 million, 50 million, 100 million is a typical size for a FASTQ file in RNA-Seq. BLAST takes a few seconds for each alignment. You multiply a few seconds times 100 million, you'll be waiting. And our computer will be churning and generating a lot of excess heat and using up electrons, and it's not optimal for all of us. We needed a faster method that takes into account big gaps. I'm not going to go into too much detail on the inner workings of the TopHat algorithm, but it finds the gap. It breaks your read into small chunks called seeds. It takes the first 25 bases, and it looks at an index of all the 25 base chunks of the reference genome and tries to find a perfect match, lacking that, the best match, lacking that, a shorter match that perhaps will run into a gap. When it finds a shorter match of some useful length, 10, 12, 15 bases, and then what looks like a gap, many bases that don't match, it jumps forward and slides along looking for where the alignment picks up again. It preferentially looks for breaks that occur at consensus intron splice sites. You can put in a command line modifier for TopHat to ignore this preference for intron splice sites at the expense of the whole thing running much, much slower, because now it has to check every single possible alignment instead of jumping to the next GC and the next GC and the next GC, which is obviously quite a bit faster, especially in an indexed genome where it can jump to all the possible reads that are downstream that start with GC in a single step. So it maps the reads to the genome with Bowtie, which is an ungapped aligner, collects all the initially unmappable reads, the ones that don't map entirely within a single exon with very few mismatches, generates possible splices looking for the next place where the next exon picks up, and then it basically creates a transcriptome, a novel version of the genome that connects up exons and then does an alignment of your read to that intron spliced out version of the genome. Okay, so that's all I'm really going to say about TopHat. Just that it's a spliced aligner. It uses a Bowtie index, and Bowtie is yet another piece of software that gives you a huge speed up compared to BLAST. And that's all I'm going to say about Bowtie, although it's relevant because you'll see Bowtie in some of the commands that we're going to type. So before you can launch TopHat, you have to launch Bowtie. And for various reasons that I don't understand, it's not possible to just let TopHat launch Bowtie all by itself. So you have to first module load Bowtie, and then module load TopHat, and then run TopHat. And you have to do that in your script like this. So again, this is showing it command line version. In a second, I'm going to show you an SGE script. Then I'm going to take a little break and have you actually type this and try and run it yourself and generate the whole world of possible errors so that you can get a little handle on how you troubleshoot. So in your script, you're going to say module load TopHat. Before that, you're going to say module load bowtie. I should have put that in here. Then you type TopHat. If you do it like this, what I really wanted to show here was for any piece of software that you're learning to use in a Linux environment or anywhere else, but particularly in a Linux environment, the very first thing you might want to try is to load up the module, read the module help. So you would say module help TopHat. In this case, it's not super helpful. Then just type TopHat interactively with no command modifiers, and very frequently the software will give you a little piece of information. In the case of TopHat, it gives you a very extensive help file if you just type TopHat with no information. And one of the very first things that it will tell you is what they call the usage, which is the correct format for a TopHat command. And this is what we're going to work on in a second. So a TopHat command should say TopHat, then a series of options. Options are command flags, so it will be a dash with a letter of some sort, often followed by some parameter, numerical or word type parameter. Or in some cases, you give it a dash, a letter, and then give it a file. And it may be that you're giving it a file that has a long path name. So that is part of the process. So TopHat, all of its options. Then you have to point to something called a bowtie index. In our case, that's going to be in iGenome's root, Homo sapiens, etc. I'll show you where that is in a second. Then you load the reads. In this case, we're just going to load one file, one FASTQ file, and map it. If you had run a paired end, you could load both pairs at the same time. And TopHat actually gains some power of mapping when it knows that two reads are near each other. Sometimes it can distinguish a read that's repetitive at one end, but matches to unique portion of the genome in the other end. And then that's it. And then you would hit return. So if we were to do this on the command line, it would look like this. But wait, don't submit this command. This is too computationally heavy a job to run on the head node. We're going to run an SGE script for it. Okay, so this is the script that I want us to start with. And I'm going to break out of here and show it interactively and show you how to look for yourself in iGenomes. There's just a couple of other things I wanted to point out about how I wrote this script before I start showing it in the terminal. All right, you remember the pound bang bin bash, pound $s bin bash, pound $CWD. We put that at the head of all of our SGE scripts so that it knows we're running in the bash shell and it puts the output in the same directory where we type the commands. All right, then we're going to load iGenomes. We're going to load bowtie, this specific version. We're going to load TopHat, this specific version. Now we're going to type a TopHat command. These backward slashes indicate that it's a line break. You can type them or ignore them and just let the line breaks fall where they want on your terminal. But we're going to do TopHat -O, and the -O refers to the output. And I'm putting a variable here, a $1. This is really a useful trick because if I put the output and the input as variables, I can reuse the same script over and over again, and I don't have to rewrite my script every time I want to analyze a new data file. I find that to be extremely helpful. So instead of putting the name of the output file here, I'm just going to put $1, and I'm going to give the output name when I submit the job on the Q subline. -p n slots, Eric told you about that yesterday. That's how many processors am I going to use. I'm also going to decide how many processors to use when I submit the job on the Q subline. And now I'm going to do -G. In TopHat language, -G means use the reference genome only. Don't try and figure out alternative splice sites. Learn the splice isoforms from the reference genome. There's another option called -g, which says use the reference genome and also learn new splice isoforms. So that's a little slower, but it does give you more expression for certain transcripts that fall outside of the known genome annotation. And if you've spent any time at transcriptomics, you know that there are alternate upstream start sites, alternate downstream transcript termination sites, and you'll accumulate more reads associated with each sample. There's a bit of a downside to that, which could be an upside, which is that some samples may have different isoforms than others. And so that will bias to some extent the overall per gene expression values, but you could dig into those isoform-specific samples and have some truly interesting discovery that a particular treatment invokes a particular isoform. So not limiting to just the known isoforms might be your best method. There's a third way of running TopHat, leaving out the G command entirely, not using a reference annotation, and allowing TopHat to figure out all the possible isoforms that your RNA might make everywhere on the genome without any regard to known genes. That will take more compute time and will almost certainly lead to differences in each FASTQ file that you align, just based on sort of random sampling issues, that it will think that there's a novel isoform here because it has a couple of transcripts that align in a slightly different way. And you'll have to massage the data more at the end of the analysis process. A lot of people don't want to do that. They don't want to discover novel isoforms that have very few RNAs associated with them. They'd like to know the expression levels of known genes. So we stick with the -G for most of our people who just want data that's compatible with what's already known about the genome. After the annotation file, then we need a reference genome. That's the index. That's back here, the bow tie index. So this is out of the options section into the required section. If you don't include a path to the bow tie index, TopHat will fail and give you an error message. After that, I've put $2. $2 represents the data file that I'm going to analyze in this run of TopHat. Again, by not putting the name of the data file into my shell script, I can reuse the same shell script with different data files. So once I've written this shell script, the only way to test it is to try and run it with real data. So now we say qsub -pe threaded. We give it a range of processors that we'd like to use. In the real world, you'd probably try more processors than this, but if you're all submitting this job at this minute, I think we ought to be a little conservative. I'm not guaranteeing that this job will get done in the next 10 minutes for all of you if you all submit it right now. Eric is frantically monitoring the cluster status to see if we crash it. So that's fine. So if you all submit with just 2 to 4 processors, the key thing that you, all right, and then we need an output file name Oh, 2 to 4 processors, then the name of the script. I called it mine, tophat.sge. You can call yours whatever you want, but put the name you made here. Then the name of an output file. This becomes $1. It's the first thing on the command line after the name of the script. That's always $1. Eric wrote some nice scripts where he put a line here where he assigned $1 to a variable name, such as out or output directory or something like that, or dir. So you could do that, and then you would use that variable name in your script. I just stuck with $1 and $2, which are the automatically assigned variable names. All right, and then after this, there's a little space there. You see that? And then this is the path to a sample name. Actually, I think that we would prefer using a path that includes RNA-Seq 2016 here, but the file still exists on this path. I didn't move it or delete it. So this is JZ5, chromosome 19. It's a human sample, but it's been subsampled down to only include those reads that we know map to chromosome 19. There's 2 things that become obvious. Number 1, the data file is smaller, so it'll run faster. 20 times faster, maybe 50 times faster, since chromosome 19 is a small chromosome and has relatively few genes on it. So it will run faster than a real FASTQ file that contains the whole genome. Also, we know it's human. Also, when we do the downstream analysis, if you were to do like a GSCA or a geo analysis to say, what families of genes and pathways are overexpressed, it may not work very well because we only have genes from chromosome 19. Also, if we load the aligned reads into a genome browser, we should only see reads for genes on chromosome 19. The other chromosomes should be pretty empty. One more thing that you'll see is since we already aligned these to the genome and then only took those reads that align to chromosome 19, you're going to get an unrealistically high alignment percentage. Nearly every read is going to align because we already know that these reads align. In a real RNA-Seq data sample, you never get more than 90% of the reads aligning, and even 80% is not a huge cause for concern. There's long non-coding RNA, there's RNA editing that can mess up the bases so that they don't align sufficiently well for bow tie to find them matching, and there's just unannotated junk in the genome that we're not going to map very well. Also, there's certain reads that come from, say, insertion sequences, ALUs, that map so many places that bow tie throws them away. So you never get 97.5 alignment percentage in real data, but that's what you'll get here. So if you write this, Q sub it, hit qstat, and it runs. In other words, you get a qstat R, and you qstat again 30 seconds later, and it's still R. You're very successful. Mostly when I do this, that never happens. What I typically see is a lot of error messages. So I usually see stuff like this. I see cuffdiff, SGE, PE error. And in addition, my Q sub disappears from the queue very quickly. That's probably because I had a typo in my script or in my Q sub. And then you go back and you try and find that typo. And that's debugging, and that's the majority of time doing bioinformatics work is spent debugging. Any kind of computer science, any kind of programming, the majority of time is spent debugging. Okay, so now I'm in the login environment, and I wanted to take you through iGenomes on a little tour before I actually submit this stuff, okay? So if I say module load iGenomes, like all good Unix commands, it gives you no feedback whatsoever. It just brings me back to the command line indicating it worked successfully. Module show iGenomes. And what iGenomes does is it sets an environment variable called iGenomes root, which actually points to a place called Phoenix iGenomes. If you type module help iGenomes, oops, it tells you pretty much the same thing, only in Eric's words.
I'll tell you where it came from.
Yeah. It tells you what iGenomes does. It comes from Illumina. Illumina gets it from UCSC. UCSC gets it from GenBank. And every step of the way, the formatting is changed very slightly. So that's what it is. The advantage is that we can update from Illumina, say, every 6 months or so, and get updated versions of all the different genomes that are supported. Also, we have pre-indexed versions for Bowtie and BWA and a couple of obsolete tools that we don't use anymore, but Illumina still supports. So you have to use this variable called $iGenomes root. So what happens if we have a look at that variable? Oops, that really doesn't work. I guess copy-paste is not so happy here.
Sadly, LS doesn't work over this.
There we go. iGenomes root. Okay, so that is a listing of what's in iGenomes root. Obviously, it's by, yeah.
So why do you need the variable to point to the file path? Why can't you just point to the file?
Yeah, that's a really super annoying thing. The file path changes depending on where you are in the system. The head node sees it differently than the worker nodes, so always use the variable name. Sorry, can't be changed. So I like the up arrow. If I say H tab, now I'm looking at the Homo sapiens. You'll notice that it automatically changed $iGenomes root to Phoenix iGenomes. Also one of those quirks of the system. But now if I list what's in Homo sapiens, we have 3 different versions of the human genome. Up to you to choose. In my example, I use the UCSC version because for my bet it's the cleanest, but if you can use the ensemble version, which has more genes, but many of them are hypothetical or even known pseudogenes. You can use the NCBI version, which has slightly fewer than the UCSC version, about 95% as many as the UCSC version. So up to you. I'm using UCSC, tab complete. So what's in UCSC? 3 versions, HG 18, 19, and 38. I ran this on HG 19 because I made this a couple of years ago, and this is where 97% of the reads align. It'll work on any of the others. You're doing the alignment now, so most of the reads will map to any genome. It's up to you. I used HG 19. So here I have to type it out explicitly. And now we have 3 different subdirectories. I'll look at the sequence subdirectory first. We have a bunch of different versions of the genome sequence for humans. We have abundant sequences, which you could use as a filter if you want to. It's not part of my workflow today. We have a Bowtie 2 index. That's what we need for today. There's a Bowtie index, which is sort of an older version. Doesn't work with TopHat versions since about 2010. So don't use the Bowtie index. You have to use the Bowtie 2 index. Even though you can module load Bowtie, and nowadays it'll give you Bowtie 2. Last year, if you module load bowtie, you got the old version, and not bowtie 2, you had to module load bowtie 2. As Eric says, the best way is to module avail, see all the versions, and use the exact version that you want to use in your script. It makes it reproducible. There's also BWA index, which is another software that's not usually used for RNA. And some over here, there's a whole genome FASTA. If you have your own tool and you just want a clean version, human readable of the genome, you can grab that whole genome FASTA and do whatever you want with it. Okay, well, what's in the bowtie 2 index? Stuff that you don't need to know, but they all start with the word genome. So TopHat, in its wisdom, likes you to point to the bowtie 2 index / genome, which is sort of the base name of all those index files. And that's what we're pointing to here. If we go back and out of here, and we look not at the sequence section, but at the annotation section, there is a section called genes, and I think that's a directory. Nope. Yep. So in genes, there are a bunch of files, and I'm not going to go through all of them, but I'm going to show you genes.gtf because I think that's interesting. So this is a format called GTF. It's related to a bed file. I'm not going to go through all the details, but as you can see, it has exons and it has positions on chromosome 1. And it also contains GenBank names and gene names. And those gene names are what will show up if you use this annotation to do Cufflinks-based gene counting. So at least you know now where those names in your output file come from. And if you don't like the names, then use a different annotation or a different version of the genome to get names that are more to your liking. These files are here, but if they're really not giving you what you want, you can come up with your own genome version, index it yourself, your own annotation that corresponds to that genome. Keep it present in your working directory and point to that instead of our reference. This is particularly important for messy genomes that are not like the big three of human, mouse, and Drosophila, where different versions may exist from different lab groups or different databases, and you may not love the Illumina UCSC version of your genome. Okay, so this is where we are. Other place I wanted to show you was the tutorial section. I gave you all some sample data in a place called, actually we'll just list ls/ifs data tutorials /RNA-Seq 2016. And located here are two files, JZ5 R1 chromosome 19 FASTA and JZ7 R1 chromosome 19 FASTA. And if we align both of those to the reference genome with TopHat by using the SGE script I just gave you, you will get two alignment output files. Rather than run this right now and wait, like Julia Child, I have pre-baked the results this morning, and I have them for you in a directory called TopHat. So if we just go to TopHat, you'll see I have two output files. I showed you in the script, I think I said JZ5 out as the name of the output. when I ran it this morning, I just called the output file JZ5 and JZ7. Yeah, nope, right. So we'll look at the first one. So what's inside of there? This is the output that TopHat generates. It always generates the exact same output with the exact same file names. The only control you have is in the name of the directory. So be a little bit careful when you specify the directory name in that qsub command so that you can sort out your output later. You might eventually rename some of these files as you use them in downstream applications. Accepted hits.bam is maybe not the absolute coolest file name you've ever seen, but that's the name of the TopHat output file. If we look at that file,
Mine only says prep reads info. Mine only says prep reads info, nothing else.
really? Are they not readable?
Here, check run your same ls command with ls minus no.
Yeah, okay.
Oh no, I wasn't different folder, don't worry, Stu.
No, we'll fix it. They're me, but I'm logged in here as demo. But the group is RNA-Seq, right? So they ought to be.
No, ls minus al?
I see it with LS.
Yeah, I see it. Yeah, no, I'm just asking for the flags just to see if there's anything funny with permissions. But really,
There's always something funny with permissions.
I mean, if he's just seeing one file, though, I mean.
No, I'm in a different directory, so it's okay.
Oh, you found it. It's not us. It's you. Well, it's always about 50-50, our error, your error. It's not like Microsoft where it's always your problem, never their problem. I'm willing to believe I messed up because I did this morning when I had, X amount of time, or X minus 5, really. So I'm doing the best I can. Okay, I was going to show you the accepted hits. That's not happening. All right, so if you look, oops, no such file or directory. Yeah, sure enough. All right, so that's what it looks like. It's a mess. Why? Because TopHat outputs files in binary alignment format, BAM. There is a tool called, oh wow, mental blank. SAMTools, sorry. There's a tool called SAMTools, which you can use to convert a BAM file into a human readable SAM file, which shows you the alignment of each read at each genome position. That's not super helpful because it's millions of lines long. You're not going to be able to make any sense out of it, and it takes up significantly more hard drive space. And all the other tools that we need to use work with the data much more efficiently in that BAM binary format. So we're going to leave that alone and just accept the fact that that accepted hits.bam output from TopHat contains our alignment result. And we're going to rely on other tools to read it. Other things in that file folder that might be interesting are the where's my TopHat output? Maybe it's in logs. TopHat log. Is that going to give me my output, or maybe it just got lost in the going? Usually, you get a TopHat report that tells you how many reads aligned. Should be there. Oh, you know what? Let's look somewhere else. There we go. Align summary text. So when you run TopHat successfully, this was from this morning, it gives you this little report. Your input in that file was a mere 1.7 million reads, not very impressive. You really need about 40 or 50 million reads to cover the whole transcriptome, but this was only chromosome 19. So chromosome 19 is sampled pretty deeply. Mapped 97.2%. So that's very impressive. Again, it's not so surprising because we only took those reads that we already knew mapped to chromosome 19, and 2.7% of those reads map in many places, and 327 of them map in more than 20 places on the genome. So those are your transposons, etc., your repeated sequences. Some of them might just be polyAs and polyTs that map all over the place. Others are ALUs and that sort of thing. Okay, so that's the output that you should expect to get if you have Q-subbed a TopHat job. You should get an alignment summary like this, and you should get an accepted hits.bam output file, as well as these other files, deletions, insertions, junctions, unmapped, which is really interesting sometimes. TopHat, I don't usually do this, but with other situations, you could use an aligner to clean out contaminants. So you first align to bacteria or E. coli or whatever. Anything that shows up in the unmapped file doesn't map to your contaminants. You rename that and realign it, and that becomes your input, your cleaned input data. Or alternately, if you're looking for the contaminants, they might show up in the unmapped file. Okay, getting back to my script. So this is your TopHat. There's two workflows after you have BAM files. There's two choices. First choice is to use a tool called Cufflinks. And for each individual sample, which is now an individual aligned BAM file, you count the number of expression level for each of the genes. In addition, there's an even more complex workflow, which if you don't want to use an annotation file, you want to predict the genes de novo, you can run a Cufflinks for each file not using a reference annotation, just making up the genes as you go along, and then merge them together for each sample so that you get roughly the same genes, plus merge in a reference annotation. So now those genes which overlap a known gene get a name, and those genes which don't get a name, plus you get a ton of novel isoforms that weren't considered in the reference annotation, because RefSeq only annotates a small number of isoforms for each gene, whereas in reality there's a huge mass of isoforms, some which differ only by a couple of bases. So those are all more complex workflows that I'm not going to show you, but if you did want to run individual samples by Cufflinks and then merge them together, this is the Cufflinks command. Again, you would module load Cufflinks, you would Cufflinks, you would specify the number of processors. In this case, I'm using igenomes genes.gtf to map to known genes, and I'm mapping the BAM file. I'm not saying which one here, so this is an example workflow, not an actual workflow. Instead, for today's tutorial, what I want you to run is cuffdiff, which is actually a separate command that's part of Cufflinks. It does the mapping of the reads to known genes, and it does the counting of reads to known genes using the annotation map, and it does the comparison from one sample to another with a statistical test all in one step. So it's essentially a script that runs a bunch of tools in series to generate differential expression values rather than single sample expression values. It actually, in its output file, shows you the expression for each of the input samples as well as the differential expression, and it also runs a statistical test to do multi-testing correction for that differential measurement. This is very widely used. You'll see a lot of publications based on the results of this script. There's been a lot of discussion in the bioinformatics community whether they use the appropriate statistical model. Most of the criticism is that it's not sufficiently stringent. In other words, it calls quite a lot of differentially expressed genes, maybe some that by rights should be false positives. That's a detailed conversation for another time. I think biologists favor this because it's nicer to get output from your experiment rather than saying you have 1 or 0 differentially expressed genes at a high threshold. We can say there's 150 differentially expressed genes at a moderate threshold. So that intuitively is more satisfying. And of course, you could always raise the threshold yourself by setting the p-value higher. So cuffdiff, same basic syntax as cufflinks. Cuffdiff, set the number of processors, set the name of the output directory, set the GTF file that has the annotation, point to an accepted hits.bam file as your input data for cuffdiff, and a second one. So here we put in JZ5 and JZ7 BAM files. So cuffdiff is always expecting 2 input files. Actually, it's more happy if you give it several input files for 2 different treatments, in other words, biological replicates, and you separate the replicates with commas and the 2 treatments with a space. So you could have JZ71, comma, JZ72, comma, JZ73, space, JZ51, JZ52, JZ53. And it'll churn more happily and give you statistically more valid results if it has some replicates for its statistical model. Otherwise, the statistical model is pretty good. So here's the example of putting that into an SGE script. I think this will work, at least it worked for me this morning on this data. I also put this script in that tutorials/RNA-Seq 2016 folder. It would be much more beneficial to you to write the script for yourself so you can experience the full range of typos. But if you want to have a successful outcome for the day, you can take this script, run it on the 2 BAM files in that TopHat folder, and get an RNA-Seq differential expression output from the day. After you've written the script, you submit it with a qsub PE threaded, the name of the script. Mine is called cufflinks.sge, and then the 2 output files go here. You'll notice I put them as $1, $2 here. So in other words, this script can be recycled for different data files. All you have to do is point to them. This script is specific for human, HG19. So you would have to edit the script if you wanted to use it on a different organism or a different build of the human genome. And this would be your result. I also put the result in a folder called diff in that same RNA-Seq tutorials 2016 or ifs data tutorials RNA-Seq 2016 diff has this output file, so you can see the format of what you're going to get. So you have a gene name, the location of that gene on the chromosome. They're all surprisingly on chromosome 19, which is cool. It sort of reinforces, because we didn't tell TopHat to use chromosome 19. We only just know that all these reads will map there. Then it has the value in sample 1, the value in sample 2, a t-test p-value, a q-value, which is a multi-test corrected p-value. It should be a larger number. In other words, it's less significant after multi-test correction. And then CuffDiff has this nice little feature, if you're an Excel user, that it flags each gene as being significant or not significant. I think I sorted them so that the yeses all come to the top, so that you have that nice warm feeling that, yes, we can find differentially expressed genes given these two very different data samples. I don't know what to say about what you should expect. Obviously, it totally depends on the samples that you submit. Different cell types will give you big differences. Lots of genes will be differentially expressed. Cultured cells with a single gene knockout may have very tiny number of differentially expressed genes. The gene you've knocked out and whatever's directly downstream from it ought to show some differential expression.
So in this case, you're submitting 2 samples.
Yes.
How is it calculating this?
Magic. Totally invalid magic.
Okay. That's what I figured.
But the fold changes are real.
Yeah, sure.
But you can't publish 2 samples anyway, so the fact that the statistics don't mean anything, they have rigged this stuff so that instead of giving you divide by 0 errors with 2 samples, it runs through the math anyway.
So if you submit a series of samples for 2 groups, it'll be a more valid.
Much more valid. Real statistical and publishable statistical. Although, as I said, there is disagreement, but no one disagrees so strongly with TopHat that they would say your results are invalid. They would say they might be a little over-rich in false positives. Yeah?
Dude, is there a way to convert SRA files to FASTQ files?
Yes.
How do we do that?
There is a SRA toolkit already installed on the cluster, and I can send you a set of instructions. It was an exercise in my class last week. It's not hard. It's a one-command thing. Yeah.
If one gene has several copies, how to identify the expression level of each copy?
You mean the multi-gene family, basically?
Yeah.
Well, TopHat does its best to identify which gene each transcript comes from. If the copies are so identical that for whatever length of read you're doing, let's say 100 bases, there is 0 difference between the 2 copies of the gene, TopHat will guess. It will map the gene to one place or the other, and it will actually give a lower mapping quality score to that gene, meaning it maps in more than one place, but it will still count it as expressed here or there. If the mapping quality score is too low, meaning it maps to, say, 5 places identically in the genome, then it probably won't pick any of those places, and that gene will go into the unmapped bin, or that read will go into the unmapped bin. You may still get a differential between the different members of the family because some reads probably have differences. There's probably not 100% sequence identity over the entire transcript. There's probably some bases that are different. A couple more things I want to show, even though I'm out of time. The accepted hits.bam file is correctly formatted for viewing in a genome viewer, such as IGV. You can download IGV from the Broad website, or just go there and hit the launch button, and it will load itself using Java Quick Launch.
Blocked by in-way firewall.
Well, download it and run it. The download isn't blocked.
Well, download is what?
Everything is blocked?
Yeah.
Eric? Can we host a copy
For IGV?
of ITV?
I've been able to get it on my system.
Yeah, me too. Never seen it. Problem. This is new. Yeah, well, today they've reconfigured the firewall. Today, because yesterday it worked. Can we host a copy of this so that people can have it?
A copy.
Of IGV, not the quick launch, but the actual jar.
So you're asking, could we install IGV?
No, can we host a copy of the download that they could get from some website?
Yeah.
We'll work it out. We'll post it on the wiki. Here's an alternate path to download from within the firewall, because we know this software is safe because we use it every day. That's a bummer, because I was going to have you all download it right now, but if that's not going to happen, it's not going to happen. Just last thing, I got 10 minutes, you can't go yet. IGV requires one additional file. It can't just view the BAM file all by itself. You have to index your BAM file. The whole point of IGV is it does not load the whole gig of genome data and visualize it all at once. That would be too slow. It would take a long time. It would kill the RAM on your machine. Instead, it zooms way out and shows you no data until you pick a gene or a chromosome region. Then it jumps to that region, uses an index to grab only those reads that overlap that region, and then it draws them on the screen. You have to build the index. For reasons I don't know, it does not build the index itself. We use SAM tools to make the index, and these are the commands that you would do. It uses moderate compute power, too much for 60 people to all do it on the head node simultaneously. So instead you put it in a shell script. It's super easy. All it does, all you have to do is SAM tools index file name. So typically you would go to TopHat, JZ5, accepted hits.bam, and then just type SAM tools index and the name of that file. It creates in that same location, hopefully, a bam.bai file, the binary index, which then you load, you copy the BAM file and the BAI file from the server onto your laptop or desktop. And then you go into IGV and you use the file load from file and point to the BAM file. It sucks up the BAM file. You may have to tell it what genome you're using, but you don't have to load a genome file. It has lots of genomes, way lot. So pretty much any genome that you're likely to use unless you're working with a custom genome you just built, like human, mouse, C. elegans, Drosophila, etc. So you may have to tell it what genome to use. It automatically sucks up the BAI file and uses it as an index. You don't have to tell it about that. And then you get a visualization that looks something like this. And then you type in one of those genes from your CuffDiff file that was known to be differential, and it should look something like this. A lot of reads here, few reads here. If you zoom way in, you'll realize that the reads actually only map to the known exons, and then these little wavy blue lines that connect the splice junctions across the introns. And if you zoom way, way in, you can actually see the individual bases. And it will highlight those bases in your reads, which differ from the reference genome. Expecting the fact that you're going to see 0.5% random sequencing error, you may see a number of positions where your read differs from the reference genome. This could be a true allelic variant. Why not? There's more than one sequence in the population. It could be 100% different, meaning your sequenced sample is homozygous for a different allele than the reference genome, or it could be something in the 50-50 range, meaning that your sample is heterozygous, or you may see something weird like 90-10, showing allele-specific expression, and you already made a discovery in your first day of genomics. Thanks. Any other questions?